The overall goal of this procedure is to generate and quantitatively analyze the elemental composition of monodispersed microdroplets containing single cells or aqueous solutions by ICPMS. This is accomplished by first fabricating a microfluidic chip using replica molding with PDMS. The second step is to connect syringe pumps to the microfluidic chip. The pumps will supply the aqueous sample and the highly volatile and immiscible organic carrier phase, PFH. When the syringe pump is turned on, the microfluidic chip generates monodispersed droplets of the aqueous sample solution or a cell suspension by flow focusing with PFH and ejects these droplets with additional PFH from the chip as a liquid jet. The final step is to insert the chip into a transport assembly consisting of a cartridge heater that evaporates the droplets and a membrane desolvator that removes the solvents prior to the ionization and detection of the droplet content by an ICPMS. Ultimately, the recorded signals and a calibration measurement with a standard solution are used to determine the elemental composition of the sample solution or the cells in the suspension.
The main advantages of our microfluidic technique over existing methods are the adjustable droplet size, the low sample consumption, and the low risk of nozzle clogging when working with cell suspensions or concentrated salt solutions. Furthermore, the low-cost disposable microfluidic chip avoids cleaning time and eliminates cross-contamination. Demonstrating the procedure will be Olga Borovinskaya, a PhD student from Professor Günther's laboratory, and Pascal Verboket, a PhD student from my laboratory.